Good afternoon, everyone. Honorable Vice Chancellor Professor R.K. Kohli, respected deans, head of schools, faculty colleagues, and my dear students. On behalf of Amity University, Punjab, the Chandigarh chapter of NASI, and SWISTI, I welcome you all to today's exciting talks on 2024 Nobel Prizes in Physiology and in Literature. We have with us two esteemed faculty colleagues, Professor Jagmohan Singh from Amity School of Biological Sciences, who will speak on the Nobel Prize in Medicine, and Dr. Pallavi Goswami from Faculty of Languages and Culture, who will speak on the Nobel Prize in Literature. Before we begin, I will request Professor Kohli, Dr. Jagmohan Singh, Dr. Pallavi, Dr. Anil Sharma, Dr. Shivali Dhingra, and Dr. Jaspreet to kindly water a plant as a symbol of growth, prosperity, and nurturing spirit. Yeah, everyone. The Nobel Prize in Medicine this year has been awarded jointly to Dr. Victor Ambrose from the University of Massachusetts, Worcester, and Dr. Gary Rufkum from the Massachusetts General Hospital, Harvard Medical School for the discovery of microRNA and its role in post-transcriptional gene regulation. This prize recognizes the groundbreaking work that has pushed the boundaries of our understanding of life, health, and healing. This achievement is not just a milestone in scientific research, but a beacon of hope for millions worldwide, reminding us of the transformative power of research. To deliver a talk on this groundbreaking discovery, we have with us Professor Jagmohan Singh, Professor Emeritus, Amity School of Biological Sciences. It is my honor and privilege to introduce Dr. Jagmohan Singh. I have known him for more than 2 decades since the time I joined for PhD at IMTech Chandigarh. Professor Singh is a fellow of National Academy of Sciences. He is a distinguished biochemist and educator. He holds BSc honors and MSc honors from the Department of Biochemistry, Punjab University, and earned his PhD at the prestigious prestigious Indian Institute of Science, Bangalore. His extensive academic journey includes postdoctoral fellowships at the University of Calgary, Canada, and the National Cancer Institute, USA. Dr. Singh joined CSIR INTEC in 1994, contributing significantly to the biochemistry and microbiology research until his retirement as chief scientist in 2017. Currently, he serves as emeritus professor at the Amity University, Punjab, sharing his expertise in biochemistry and advancing scientific education. With over 75 research publications and numerous citations to his credit, Dr. Singh's contribution have shaped our understanding of epigenetics, heterochromatin, and other critical areas of molecular biology. Please join me in welcoming Professor Jagmohan.
Thank you, Dr. Vishal, especially for this nice poster. For once in my life, I haven't seen myself the same thing. As Vishal has told in the introduction, so many of our discoveries are made when we reach a stage where we don't know what's going on. We have an observation and we don't know, we cannot explain it by conventional dogma. So I'll start from there and I'll go through 2 steps through which these 2 different discoveries leading to this discovery we're talking about took place. Um,
Make it simple.
Yes,
So
yes.
that non-biologists,
Yeah.
they can understand.
I'll try. So, title is that most of our understanding of biology is that all things are made by proteins, and proteins are encoded by messenger RNAs, and that's how it works. But now picture has changed drastically with the small RNAs which control everything. So this is the repeat of the previous slide. Ambrose and Rumpel got the Nobel Prize for discovery of microRNA's role in post-transcriptional gene regulation. So important key point here is post-transcriptional gene regulation. Earlier understanding was only trying to explain transcriptional gene regulation. You have some new organism, new tissue during development, you make RNA corresponding to that, like tissue-specific protein. So going back to the background, our genome can be likened to a pointer to basically starting from a single cell, whole body comes. Any organism you take, it's happening. So the blueprint is in the genome, where all the information is there, and then those are genes are carrying instructions to make proteins, and which are typical for various cell types in the body. So it is visualized in this picture that conventional dogma was DNA makes mRNA, mRNA makes protein, and each cell type has specific set of proteins: neuronal cells, muscle cells, epidermal cell, gametes, and so on. So center dogma was like that until things started looking puzzling. This is just a prelude to what happened. This strange phenomenon was known called as position effect variation. We have the same person. Those who are a little bit into gardening. We were familiar with this picture in our house plants. One would say, think, यार ये है तो green है pigment, लेकिन कुछ parts of the क्या कहते हैं, leaves are not having color. What's happening? Genetic makeup is the same. So this was broadly explained as epigenetic process. But epigenetic is a very wide process. Some parts of the leaf is expressing a particular pigment, others are not. How? What? And this experimentally observed in Drosophila, and it was linked to normal red eye color of Drosophila in wild type changes to variegated. We have that in mind. So it was custom function was assigned to this locus called W, white locus, and then it was found that in the variegated eye there is an inversion of the temperature. So basically, a pigmentation gene is being either expressed or not expressed because of the locus. But this was simple comparatively. Until somebody tried to again play with the nature, petunia plant. In the 1990s, this group tried to, they identified the gene for purple pigment, They said, we want to make beautiful purple flowers. So let's overexpress the gene. So one would expect you get more variegated, more sort of stronger variations of purple pigment. Unfortunately, they found that outcome was totally opposite. They saw total loss of color. So pigmentation, in other words, charcoal synthesis, instead of being made, was not being made. What's happening? So the solution came from a C. elegans system. It's a small worm, 1 millimeter long. It has been widely discussed, researched, and its genetic and developmental program is very well known. And today's discovery is also built on C. elegans. So what this group earlier worked, Fire and Mellow, what they thought was to understand how the expression can be enhanced. They were not even probably thinking what they are addressing. Idea is to knock down the expression of a gene which you can follow by either antibody, it gives a dark color, or you can have a fluorescent labeled dye. So you can visualize the product. So they said, okay, we want to knock down this gene. They put antisense on it. That was the field, latest thing in the field at that in 1990. And they said, no, but it didn't cause partial, it only caused partial loss of signal for MEX3. Then one of the control experiment, they accidentally mixed up sense and antisense protein. And they got the surprising total knockout of the expression. That made them thinking. I think from there they said, no, we have to change our paradigm. So these are the two guys who got the Nobel Prize for the discovery of RNA interference, gene silencing by double-stranded RNA. Now, so this is the timeline. The earlier purple color experiment was done by Pinoli et al. Called as co-suppression, 1990. 98 gave the initial insight, where what is the underlying possibility. And quickly, in 1999, they found that there are small RNAs, short, आजकल उनको बोलते हैं SIR, small interfering RNA. And the gene which was responsible for that was tyrosine. Then this phenomenon was discovered in wide variety of species, even a simple organism like fish and yeast. And that, because of ease of doing genetics and biochemistry with fish and yeast, some of the underlying mechanisms were discovered. And broadly speaking, what happens is the double-stranded RNA we are putting into the cells gets cleaved by dicer into small interfering 20-25 days long RNA. Then subsequent players, what they do is they remove one of the strands as called as passenger strand on the left side and keep the other RNA, single-stranded RNA, in a complex form. That complex goes to target mRNA. So what is happening all around? mRNA is being recognized, converted to double-strand form and cleaved by dicer. So in the process, mRNA is destroyed. Inherent because it also amplifies itself. These complexes of mRNA with a lot of proteins called RNA-induced silencing complex, etc., can make copies of fresh mRNA using this small RNA as a primary with the help of RNA-dependent RNA. So it's a self-amplifying system. So this was the precursor to this work because what is the common working element is the same in the discovery we are talking about. Ambrose and Nobel. So this brings us back to the C. elegans development. The life cycle shows it's a egg and sperm form the embryo develops and goes through these four larval stages, L1, L2, L3, and L4. Intermediate is not relevant, but due to crowding and low food, it can convert into hibernating form called dower. And it takes several months to bring back to rich medium, it convert to L4 and produces the drug form. So people have been studying a process called heterochronic genes. These refer to development of cells or tissues at an abnormal time relative to other unaffected events and serve as temporal landmark. How it was importance was discovered by virtue of mutants. In some of the mutant, development mutant, only timing shift हो जाती है. Regardless of nerve cell generation, esophagus and all this. These are minute mini organelles, unlike ours, these are huge. So only जो timing during development shifts in these lymph4 mutants. For example, in lymph4 mutant, people found lymph14. Can go back to this figure.
Because of the food, amount of food.
Yeah, in rich food, this is what's happening. But it's automatically regulated.
All things alone. So no food.
Low food will take them to hibernation. Tower.
So this will impact all other process.
Yeah, but then they can short circuit to L4 upon providing the medium. But this is the normal development in treatment.
Okay, I'm saying that in covering low
Yeah.
number of
Yeah.
larvae.
Because they are competing, they are not able to individually get enough food. They just have to go slower.
Energy related.
Energy. But what we are talking about is these two. These are tightly coordinated with synthesis of lin-14 protein. lin-14 आता है शुरू में L1 के बाद वो कम हो जाता है normal development, and at the same time lin-4 goes up. So there was some kind of coordination. Whether it was causal or then consequences is not known. But people genetically found out that lin-4 is regulating lin-14. अगर lin-14 mutant होता है जिसमें lin-14 का अपना expression, it can go on to second, third stage also. The important thing was there was an idea that at certain stage something is happening. A lot of factors are known. Yeah, I'll come to that. This is another regulated event at the end of it. So it was found, original discovery was made in late 1980s with the lin-4 discovery of lin-4 mutation. So this finding remained in hibernation without any explanation. What's causing this? So then later on, Ambrose and Rupkin studied Rupkin, I was looking at lin-14 mutants, and together they started talking to each other. And they found lin-4 is apparently negative rate. That's shown by the previous correlation, negative correlation. There's a flavor of what lin-14 does is that normal axonal, briefly speaking, axonal development, nerve cell connection between two nerve cells to axon is disturbed in the lin-14 mutant. So you see the formation of a normal PLM axon in wild type, in mutant it is disturbed. For now, I think that is enough. So before coming to the results, let me give you a flavor of what is happening here. What is the overall knowledge in the field? That DNA makes RNA. RNA is just not the coding sequence. It has 5' UTR. UTR is our translated sequence. 3' UTR. So UTR, 5' UTR has methylated guanosine to protect it and also allow entry of ribosome. And it has normal hairpin to for ribosome entry. It has another IRES, which is independent of methylated guanine. So it can recognize RNA here, which are not methylated. So these are fail-safe mechanisms to ensure translation happens. So everything was okay, mRNA makes protein, but no one paying attention to 3' UTR. 3' UTR has complex sequences. The obvious things were that you have a protein complex which ensures subcellular localization, stability, polyethylation, which again takes care of the stability of mRNA. That was where everyone stopped. Now, what they found, long story short, Victor and Ambrose talked to each other and Rupkin talked to each other. They found they sequenced lin-4 and lin-14. They found lin-4 is encoding a very small RNA. This was unusual. Everyone is looking for long RNA. Gene है, mRNA होगा, बड़ा RNA होगा, न बड़ा protein होगा. तो उन्होंने interesting thing is when they talked to each other, they found lin-4 is complementary to this, right side to the 3' UTR of lin-14. So that was the key clinching piece of observation that made them think. Then they found, studied their regulation. lin-4 is regulating its own transcription positively, and translation of lin-14 level. So here it is working at the promoter level, lin-4 promoter. All the factors which are involved in transcription are used to make more of the small RNA. तो हम माइक्रो आरएनए को वो माइक्रो आरएनए lin-14 के 3' UTR mRNA का 3' UTR पर बेस पेयर करता है, उसकी ट्रांसलेशन को ब्लॉक करता है. So proteins are being made till they are needed. फिर lin-4 small RNA आएगा, अब उस प्रोटीन लेट 14 की प्रोटीन की जरूरत नहीं है स्टेज 1 के बाद, सो उसको ट्रांसलेशन को ब्लॉक कर देता है। दैट विल ट्रिगर द डिग्रेडेशन ऑफ लेट 14 mRNA कि वो उसका काम हो गया, सो इट disappears. So this is the summary. It was a dual discovery. A novel RNA, it's called as microRNA, and previously unknown mechanism of gene regulation, that is post-transcription of gene silencing. So now these two
microRNA was known earlier also.
microRNAs were discovered by them, but it came from the RNAi discovery
Now microRNA
in
नाम
98.
को थी।
98.
अब उन्होंने इसकी सिग्निफिकेंस को चेंज किया।
Huh. I'm summarizing in this slide. So this is part where we combining the two discoveries. See how it is happening at a very simple level. Primary microRNAs are being made, we have a stem loop structure. They are processed to make pre-mRNA with dosha enzyme. That pre-mRNA is exported through a variety of proteins, but what is the critical step is action of dicer. वो only base pair region को free कर देता है, releases the so-called small interfering RNA or mature. It can have two forms. One form is where it जो हमने पहली स्लाइड में देखा, it acts if it is perfectly complementary to the target mRNA, it recruits dicer, which cleaves the mRNA. So this is one way of post-transcriptional silencing. The other is just like in C. elegans. Partial complementation, what it will do is mRNA is formed heteroduplets क्योंकि partial homology है, बीच में bunch है. So that is structure is recognized by set of proteins.
which bulk जो है ये stabilize कैसे होगा?
Other proteins bind. Actually जो flanking sequence है वो homologous, वो base pair होता है. That keeps the structure stable. But this structure is recognized by host large number of factors.
छोटा है इसलिए possible.
So this is evolution. कि सारे protein complexes have co-evolved in this process, and they block the translation. It seems like a very fascinating new odd phenomenon. Then what? जैसे आपने शुरू में पूछा, Rupkin then went on to clone LET7, which is also encoding a microRNA. And what is interesting is he found that जब LET4 was not found in humans and any other living, it was found only in CLE. But LET7 is found in all organisms, right up to humans. And if you look at the evolutionary, you can draw phylogenetic tree and trace back to 500 years. It evolved very early. जब ट्रांसक्रिप्शन शुरू हुआ तभी ट्रांसलेशन के साथ ही ये हुआ, क्योंकि in development you have to not only make proteins, you have to make at a certain time and then let them go. They trigger a process of differentiation and then disappear. Consequences of that will be seen in LET17, LET14. If you remove the 3' UTR, everything goes up. Neuronal connections are disrupted and all that. So now we know that LET7 is highly conserved during the evolution, and what that has triggered is Dr. Vishal said in the introduction, this has exploded. All these applications are coming out of it. Cancer means that thousands of mRNAs are found in humans. One can imagine वो कहाँ target करते हैं. And this can be exploited. जैसे हमने siRNA को antisense को use किया है, we can exploit this machinery to knock out the oncogenes, whichever is causing cancer. We can knock out the big oncogene, providing these. Now tools are available. You can make these mRNAs, small mRNAs in vitro, with the control of template sequencing, and exploit the host cell machinery to make the mRNA of your own choice. So at the end, I'll just summarize. All these discoveries had a very long timeline. Idea was that time, the best motivation to understand or explanation why people are excited about it is that a famous statement by Edison that about this invention of the light bulb. Somebody said you tried 1,000 times before you made it successful. He said again 8,000 times. The light bulb was an invention with thousands. So scientists are
Can they have questions?
At many and 14, are they conserving only?
link 4 is not. Link 14 is not. That's 7.
That's it. That's it.
इसको मैं
Presence of my RNA, my bacteria, or any other cell. So what would be the cost of that? Whole type of my RNA, screening for my RNA at any particular point of time during experiment. So what would be the cost if you go on? We also I don't understand your question, but I think what I can say is that mRNAs, going back to their evolution, they are present in all cells. These regulatory mechanisms were not just, they were necessary to be put in place. Apart from the normal gene regulatory mechanism we familiar with.
This question is different. He says that this micro RNA इसको identify करने के लिए cost कितनी लगेगी अगर ये identify करना?
That is sequencing, my RNA sequencing. Not very much cost.
The small sequence
sample. I see.
It is around 25 to 30,000.
Many most of them.
It's first
Basically, there are methods to fractionate RNA into small RNA and larger. Very simple. I can share the protocol.
Will they find physiological relevance is very difficult
Yeah.
because microRNAs can do even with partial complement.
Yeah. So that's where you have to reduce the stringency of the candidate. कि कहाँ partial complement. Maybe take sections of it.
So rather than identifying targets than identifying micro.
Then do two portions separately and see where they align, and then combine them together where they align together. Is it like we make RNA sequence the same way? जैसे RNA sequence में करते हैं, libraries बनाई जाती हैं, same techniques are used. Only thing is you size select the small RNA. These are standard methods. We have also done that. So not so difficult. आजकल सब processes बहुत simple हो गए. और कंपनियां आपके लिए सारा sequencing करती हैं.
Any questions from students? Can we have one question from student? Anyone? Okay. So let's thank Professor Jagmohan Singh, and I would request Professor Kohli to kindly felicitate Professor Singh with the memento.
सबकी तरफ से
So equally captivating is the Nobel Prize in Literature, which honors an author whose words have transcended borders, languages, and cultures to illuminate the human experience in profound and unforgettable ways. Literature connects us to one another, fosters empathy and understanding in a world that often feels divided. Now we'll delve into the life and works of this year's laureate, Ms. Kang Kang from South Korea, celebrating her contributions and inspiration. Ms. Kang's accomplishments remind us of the beauty of curiosity and the importance of perseverance in the pursuit of truth. To deliver the talk on this award-winning work of Ms. Kang, we have with us Dr. Pallavi Goswami, a distinguished academician and linguist with over 19 years of experience in teaching and publishing. She holds a PhD and MPhil in English from Jawaharlal Nehru University and BA honors in English from the University of Delhi. Currently, she serves as an associate professor at the Amity School of Languages and Culture and is also an associate dean of the Faculty of Languages and Culture, Amity University, Punjab. Please join me in welcoming Dr. Pallavi Goswami.
you, Dr. Shah, for your kind words. I've been entrusted with the responsibility of talking about the Nobel Prize in Literature 2024. I hope you understand that it's not an easy feat because it encompasses the work of an author, a lifelong work, I think, and I'm going to be able to talk about her in brief because I don't think so a seminar or a conference is enough to be able to talk at length about the work of any particular author. So without taking much of your time, so I'll just go on to introduce you to the person who has been awarded the Nobel Prize in Literature 2024. She's a lady, she's an author from South Korea, Seoul, and her name is Hong Kong. Right, that's how it is pronounced in Korean, as I've been made to understand. So I'll continue using the Korean pronunciation as Hong Kong. Though in English it would be Han Kang, right? So some of you might wonder, whereas there are 5 categories of Nobel Prizes: physics, chemistry, physiology or medicine, Nobel Peace Prize, and one in economic sciences, which mostly belongs to the domain of sciences. Why would there be a Nobel Prize in literature? Any guesses from the audience? Why did Alfred Bernhard Nobel, decide to establish a Nobel Prize in literature? Take a guess. Take a wild guess.
Culture. To identify different cultures. And it can be identified well through literature.
You're right, sir. You're right. But personally speaking, on behalf of Alfred Bernhard Nobel, he had broad cultural interest. So he developed his literary interest at a very young age. He had a huge library, which had a rich collection of books in different languages, literature in different languages. You would be surprised to know that during the last years of his life, he tried his hand at becoming an author. So he wrote books in poetry and drama. So his literary interest he made his mind that in addition to the other categories, he would also establish a prize in literature, and this was the fourth prize area for the Nobel Prize. So this is part of the will of Alfred Bernhard Nobel. He said that the said interest shall be divided into 5 equal parts, which shall be apportioned as follows: 1 part to the person who shall have produced in the field of literature the most outstanding work in an ideal direction. So literature, as we understand, was very close to his heart in addition to the other fields. Let me introduce you to the awardee, the Nobel Prize laureate. Ms. Hong Kong, she was awarded, and she hails from South Korea, as I said. She was acknowledged, these are the words, quote unquote, for her intense poetic prose that confronts historical traumas and exposes the fragility of human life. I'll talk more about her work. She's the first South Korean literature laureate to talk about her life. In a nutshell, I will not be able to talk at length about her life, but just to give you an idea of where she was born. She was born on 27th November 1970 in a place called Gwangju in South Korea. Her parents are Hong Sung-woon and Im Gang-ho. She moved to Seoul at a very early age from Gwangju. She moved to Seoul, where she still resides. And coincidentally, her father is also a writer. You might think that, why is her poetry? So we'll come to that. She engages also in art and music, which has strongly influenced the form and content of her writing. She writes in Korean. Most of her works, or in fact all her works, are written in Korean. And there's a very famous translator who has translated her works into English. Her name is Deborah Smith. So the works have been translated into English and over 20 other languages. So she's widely read, her works have been widely translated. She was married to Hong Yong-hee. However, they've had a legal separation, and they have a son who currently resides with her. Coming to her career, which is the most important section of my session today. Hong Kong's literary career began in 1993 with the publication of her poems in Literature and Society. This is a quarterly literary magazine, which was earlier titled Literature and Intelligence. So later it was renamed Literature and Society, and her poem, a collection of poems, was published in 1993. So initially, she started her career as a poet, but she later expanded her literary oeuvre by writing novels and short stories. So, in 1995, a couple of years after her poetry collection was published, she made her debut as a prose writer with a collection of short stories which was titled Love and Yusu. From 2007 to 2018, she taught creative writing at the Seoul Institute of Arts. Now we come to her international acclaim. She won international acclaim in 2007 with a novel titled The Vegetarian. It's a very interesting book. I hope some of you are able to read it sometime. You will understand the complexity of her work, the way in which she has tried to make it very interdisciplinary. And it became the first novel in the Korean language to win the Man Booker International Prize for Fiction in 2016. So a few years after her publication of the first novel, she got the Man Booker International Prize, which is a very acclaimed, prestigious award for fiction in English. Mainly a novel about a woman's decision to stop eating meat, which is not so simple as it may seem. It revolves around a lot of decisions which she had to take, and which led to a lot of problems, which leads to violent consequences. But there are other many layers of the text which you will unravel as you read. It's not so easy as I may seem to portray, and my humble request is to go deeper into it, which I may not be able to do at this point. So Hong Kong is seen to critique societal norms in these novels. She's a commentator, she's a critique. She has spoken about the society in which she has resided for so long, and she has protested against a lot of events which has scarred the history of South Korea. Some of you may be aware of the massacre on Jeju Island in South Korea. It's an island in the southern part of South Korea. Some of you may also be aware of the massacre in Gwangju, where a lot of student protesters were also massacred. So she's written about all this in her books in a very poetic, evocative, and brilliant manner. Again, in 2018, her book, The White Book, was shortlisted for the 2018 Man Booker International Prize, which is again a very lyrical and unsettling exploration of personal grief. This book is focused on the 1980 massacre, which took place and which shook South Korea. Right? So she has gone ahead to talk in very descriptive words about the massacre, about the history of the place, about the background, in such a way that it will leave every reader compelled to read further, not to lay down the book, not to give it one reading, but to give it multiple readings. So that is the quality of her work, that is the style of her work, that is the brilliance of her work. In her works, Hong Kong, as I said, focuses on historical traumas and the invisible set of rules that govern human behavior. All the traumas, the pain, the suffering that she has seen unfold in front of her eyes, or she has witnessed, or she has heard of. All those things have been captured in her books. And they are no doubt no less than anybody else's who would have recounted these events in a similar manner, but she has brought it out to the audience in such a way that it leaves them spellbound. In each of her works, she delves into the fragility of human life, how fragile our lives are, the impermanence of life. So she has talked about all these in her books. And at a very young age, she has understood a remarkable, just connected body and soul, the living and the dead. And her writing often reflects the delicate boundaries between life and death, body and soul, which sheds light on the fragility of human existence. We are not sure of where the boundaries lie between body and soul. We call our soul, we call our body, but what are the boundaries? We don't know, we're not aware of them. We're not aware of the boundaries between life and death. So she has looked at the connections between all these things and between human and non-human, between dream and reality. So all these are interconnected, and it's very difficult to set a boundary. It's very difficult. So she sheds light on all of these aspects. Her notable works, as I said, are The Vegetarian, which got the Man Booker International Prize in 2016. So it was published in 2007, Human Acts in 2014, and The White Book in 2016. Her forthcoming novel, if you're interested, there's a Kindle edition, which is you can pre-order on Amazon. It's available. It's for about 1,300 something, and you can pre-order, and it's going to be published in February 2025. In many of her books, including The White Book, Hong Kong tackles the deep scars left by historical events in South Korea. The White Book specifically focuses on three women. So the story is the protagonist. There are three women who are attempting to channel the pain of a 1948 massacre on South Korea's southern island of Jeju into a shared art project. So these are real events which happened in South Korea. It's not fiction. So it's portrayed in a fictional form, but it's drawn from real life. So these are real events which happened in South Korea. So many of Kang's works address grief and loss in various forms. She's always talking about the pain, the loss, whether it's the loss of a loved one in a massacre, the loss of sight, the loss of hearing or speech, or the loss of a sibling never met. Why do you think she would win a Nobel Prize in Literature? There must be a distinct style in her writing. She's particularly known for the kind of prose that she's written, the kind of poetic quality that she has been able to lend to it, marked by a lyrical and experimental style. It's very lyrical, it's very poetic. That is why the distinction lies. That is where the quality comes from, the brilliance comes from. Despite of writing about these massacres, the form that she's taken to, kind of style that she has adopted, is so lyrical, it's so poetic, that it has set her apart from all the other authors. And she's continued to do this in all her works. You would also know that there are certain institutions which have been designated for the Nobel Prizes. So for literature, the Swedish Academy has been entrusted with the task of awarding the prize in literature. So according to the Swedish Academy, her inventive approach to poetic prose, this is from the Swedish Academy, the institution which selects the final candidate for the Nobel Prize in Literature. So according to the Swedish Academy, her inventive approach to poetic prose has made her an innovator in contemporary prose.
I have a question.
Yes.
What do you think one with a matter of chance that she was born at that place, that she could see people in grief or massacre was happening? So that was the main thing, or her intense approach.
Her inventive approach, as I said. You may be born in the same place, but you may not be able to capture the events in the way that she has. Yes, but I don't think that anybody else, or too many other people living in South Korea will be able to portray the events in the way that she has been able to do. So living in one place is one thing. Yes, you have to undergo the same kind of experience, yes. But to be able to present it to a wider audience in a way which is captivating, which is riveting, is something that is touching, that's enduring, that is going to last for generations. We don't write for today, we write for generations to come so that they read our books. So we are still reading books which have been written centuries ago. We're still reading Aristotle, we're still reading Plato. There must be some significant work that they've done, right? So she has written in a way wherein generations to come will be able to read her work and understand what had happened And why is she writing in the manner that she is? As I said, her work focuses on mostly on grief, on suffering, on the pain. So I mean, it's not easy to forget such incidents. And it's not easy for South Koreans, I'm sure, to be able to forget it for generations to come. Right? So if she's writing not only for a person who's in South Korea, she's writing for a wider audience to understand the pain, the grief that they've undergone. So that is where the brilliance comes from. Yes.
I have a question about the translation. You just said that there have been 20 language translations. So in the translation, what was the approach to the political?
Yes. So the Man Booker International Prize for the Vegetarian was shared by the translator. She shared it with the translator, Deborah Smith. So the translator is equally responsible for we always say that anything in translation is lost, right? There's a loss in translation. But for a translator to be able to bring out that essence, the same essence that is in the Korean language, is a mark of brilliance for the translator. So the prize was shared by the translator. Yes. And it is fair enough that, because it's in English, it has to be shared, right? So did I answer your question?
Definitely.
Yeah. All right. So I'll just give you a little bit of excerpts from her works to make you understand how she writes. So this is from The Vegetarian. The sunlight that came splintering through the wide window, dissolving into grains of sand, and the beauty of that body, that though this was not visible to the eye, was also ceaselessly splintering. The overwhelming inexpressibility of the scene beat against him like a wave breaking on the rocks, elevating even those terrifyingly unknowable compulsions that had caused him such pain over the past year. So you can understand that she's writing about a scene, she's writing about emotions in a way which would, if you're a reader, you would be able to visualize, you would be able to imagine in front of your eyes. The way in which he's thinking, the protagonist is thinking, the pain that had caused him over the past year. In another work, Human Acts, she writes these beautiful lines. After you died, I couldn't hold a funeral. So these eyes that once beheld you became a shrine. These ears that once heard your voice became a shrine. These lungs that once inhaled your breath became a shrine. It's so poetic in nature, isn't it? It's prose, but written in such a poetic manner that it is nothing short of being beautiful. Another one, even more brilliant. How long do souls linger by the side of their bodies? Do they really flutter away like some kind of bird? Is that what trembles the edges of the candle flame? Wow, isn't it beautiful? I mean, as you keep on reading, you will just go deeper and deeper and just feel, I'll come to how she writes and how she tries to probably through sensory experiences, touch, smell, hearing. So in an interview after she won the Booker Prize, in fact, she shared that she places great emphasis on sensory experiences when writing, with an aim to evoke certain sensations of touch, hearings, sight, smell in the reader. And these sensations, the readers would feel as if there's an electric current passing through them, because they're so vivid. They make you visualize and you actually feel the sensations of what she's trying to portray. It's rather a sensory experience. Instead of just a mere reading, it actually is a sensory experience. So there is another Japanese writer called Haruki Murakami. Some of you may have read his books. So her style has been likened to that of Haruki Murakami, and he writes in a similar manner, where the boundaries are not defined. The boundaries are blurred between human and non-human, between sanity and insanity, between life and death. So the boundaries, you don't know where the boundaries are. It's almost like surreal experience. It's almost surreal. You cannot understand whether it's human, the connection is between human and non-human, or it's between the plant life and the animal life, or what, or life and death, or body and soul. It's just like such an experience when you're reading that you go into a world which doesn't seem real. Okay, now to come to an interesting fact about her reaction to the Nobel Prize in Literature. So when she got a call, so normally you just get a call from the Nobel Committee, member of the Nobel Committee, telling you that you've been awarded the Nobel Prize in Literature. So this happened on a day when she was having dinner with her son in Seoul. And I'll just read you a little bit of excerpts from the interview, from the call that she received from the Nobel Committee. So, all right, so she said, so this was a person called Jenny Ryden who had called her in the evening. So she's like, Hong Kong, hello. Hello, is this Hong Kong? Yes. My name is Jenny Ryden. I'm calling from the Nobel Prize. So if some of you may be awarded, you might also get a call similar to this. So I'm just saying, you be prepared. So nice to talk to you. Very nice to talk to you. Please let me express my congratulations. Thank you so much. How are you feeling right now? Oh, I'm so surprised. And absolutely, I'm honored. How did you find out about the prize? She's so humble. She's like, someone called me and he talked to me about this news. And of course, I was surprised. And I just finished dinner with my son. And in Korea, it's just 8 o'clock in the evening. So yeah, it's a very peaceful evening. I was really surprised. And are you in your home in Seoul? Yeah, I'm at home in Seoul. And what have you been doing today? Today, I didn't work today. And I just read a bit and I took a week. It was kind of a very easy day for me. So Jenny Radin was asking her, so you're with your son? What was his reaction to this? So you would expect that he's jumping with joy. So she says, my son was surprised as well, but we didn't have much time to talk about this. We were surprised and that's all. It's me to you getting the Nobel Prize in Literature. Yes, I'm honored and I really appreciate your support, the support of the prize. I just appreciate it. You're the first literature laureate from South Korea. How does that feel? Now she wants to ask, like, get more. What is the reaction? You're the first Korean, South Korean. Yes, I grew up with books, So since when I was a child, I grew up with books in Korean and translated as well. So I can see I grew up with Korean literature, which I feel very close to. So I hope this news is nice for Korean literature and my friends, readers and writers. Jenny Ryden comes, the next question comes. You said you come from a literary background. What writers have been your most important sources of inspiration? She answers, for me, since when I was a child, all writers have been collective. They're searching meanings in life. We all know writers write, looking for meanings, seeking meanings. Sometimes they're lost, and sometimes they're determined, and all their efforts and all their strengths have been my inspiration. So it's very difficult for me to pick some names of the inspiration. It's very difficult for me. Then comes another question in the course of the conversation. There comes another question, the last one. I will let you go in a short while. Do you have any ideas on how you will celebrate this Nobel Prize? That's the last question that comes. How will you celebrate now that you know that you've got the Nobel Prize? How will you celebrate? After this phone call, I'd like to have tea. I don't drink. I'm going to have tea with my son. I will celebrate it quietly tonight. Very nice. Yes, many congratulations again. Thank you so much. Thank you. Okay, goodbye. Take care. Bye. So this is how the conversation about the Nobel Prize in Literature 2024 ends. So you might think that, she would have so many words to express her joy and enthusiasm, but she just puts it in a very short, in a few words and says that I'll have tea. I'm going to have tea. I don't drink, so I'll enjoy a cup of tea. So this is Hong Kong. I'm sure none of us would have this reaction, right? So I'm just telling you the person that she is. All right. I don't know if I should be doing it at this stage, but since I have this opportunity, I should, yes, do it. And is looking at me and saying yes. So I think because I was only given the task of talking about the Nobel Prize in Literature, but I spoke to Dr. Vishal as well, and he said that I can talk about the Nobel laureates and nominees from India, if I may. So, yeah. Yes. So we know we have a South Korean literature laureate, Hong Kong. But do we know how many Nobel laureates do we have from India? In all fields, how many Nobel laureates do we have? We are talking about Nobel Prize, right? So I'm sure you have an idea. Six, so there's more. There's more. You can name it whichever field you are in. Nine? Do you think there are nine? Nine Nobel laureates? No? Are Nobel Puranas? Yes.
Which is last year? Economics.
Abhijit Banerjee shared jointly. Okay. So, C.V. Raman. Rabindranath Tagore. Yes.
Arvind Krishna.
Arvind Krishna.
You can put Venki Ramakrishna.
Yes, you can even put B.S. Naipaul, but he's not. Yes, it can.
Mother Teresa.
Yes.
Mother Teresa from Kolkata. Mother Teresa.
Nominees, are we talking about laureates or?
Laureates.
Mother Teresa, the Peace Prize. Surprisingly, now that we're talking about the Peace Prize, Mahatma Gandhi was nominated several times, but he was never awarded for the Peace Prize. Whereas Martin Luther King was awarded the Peace Prize, but not Mahatma Gandhi. So we have one Nobel Prize in Literature, which was awarded to Rabindranath Tagore in 1913 for his work Gitanjali.
You know, those who are Indian citizens, I am not counting those who were born in India.
Yes.
No, उन्होंने तो अपनी citizenship change कर ली.
Who were not born.
Yes, those who are not born. I don't consider them.
No, so I'm not counting B.S. Naipaul. I'm not counting it. So, yes, you're right, sir. We're not counting them. So, our first Indian Nobel laureate, Rabindranath Tagore, received the Nobel Prize in Literature for his book Gitanjali in 1913. And he was acknowledged for his profoundly sensitive, fresh, and beautiful verse, by means of which, with consummate skill, he has made his poetic thought expressed in his own English words a part of the literature of the West. Right? So, Rabindranath Tagore, obviously, although Tagore originally wrote in Bengali, he translated his poetry into English, which earned him a huge fan following in the West. And his poetry was also thought to convey the peace of the soul in harmony with nature and in contrast to the life in the West, which is frenzied and chaotic. But there's another interesting fact, I'm sure all of you would be aware, that if you're nominated, your name, and you're not awarded, you're nominated, but you're not awarded, your name will not be disclosed till 50 years later. So we don't know the names of the nominees till 50 years later. So based on the data for the years 1901 and 1964, Indians nominated for the Literature Nobel Prize, apart from Tagore, include Robi Datta in 1916, Dr. Sarvapalli Radhakrishnan 5 times between 1933 to 37, Sri Aurobindo in 1943, Bensadhar Majumdar twice in 1937 and 1939, and philologist Sanjeev Chaudhary twice. So there are quite a few nominations from India, though there's been only one Nobel laureate in literature. So it's a very interesting process, which I'll explain to you later, of how the selection is done. So it's a long process wherein I told you that these awarding institutions, they have the charge of choosing the final candidate. But the nomination letters are sent out to various universities and institutions for them to choose a candidate or other or multiple candidates. So the nomination invitations are sent out in January or in September, and then 31st Jan is the deadline for submission of nominations. So you can, so it goes to the awarding institution, the letters are sent to the awarding institution. Then 15 in April, 15 to 20 preliminary candidates are chosen. In May, 5 final candidates for that particular category. So as I said, in literature, it's the Swedish Academy. For sciences, it is the it's the Academy of Sciences.
For physics and chemistry.
Yeah, that's what I'm saying, for physics and chemistry. Then for the Peace Prize, it's a very strange fact. For the Peace Prize, the Norwegian Parliament has to nominate 5 persons who will choose the person who will get the Nobel laureate Peace Prize. So that's Norwegian Parliament. And then there is enough for economic sciences as a bank. Yes, the Central Bank of Sweden gets the number who chooses the candidate. So there are all 5 awarding institutions. Nobel himself, Alfred Nobel himself designated these institutions and gave them the responsibility of awarding.
Economics was
Not in 1968.
It was 1969.
1969. So the Central Bank of Sweden.
1901. These years there were no Nobel Prizes.
No Nobel.
And this in economics is based on the interest that in the Swedish bank,
Swedish.
based on that interest, and the amount is not equivalent to.
Yes, it's not.
Approximately.
It's not as much as the Nobel Prize. So all these institutions, they are entrusted, and he has signed his third and last will in 1895. He died in 1896. So the year before he passed away, he signed his last third and final will, wherein he established these prizes, and he also designated the institutions which would be responsible for awarding. So he had very well thought out of how these prizes will have to be established, what kind of amount will be given. So there's a medal, there's a diploma, and there's a prize money. And for each category, not more than three can be awarded. So there can be 3 laureates, but not 4 for each category. So for literature, it's only, I think, in 4 years that it has been shared jointly. Only 4 years, because literature is perhaps the only field wherein, it's your creativity, it's your way of expression, rather than a joint collaboration for a certain scientific discovery. So it's only 4 years that, it has been shared. And yeah, that's about it. So
How much is the price money?
it's about 31 million. No, he had kept 31 million. What is it called? Swedish krona. So 31 million was left kept aside for this. So
Currently it's 2.2 billion.
2.2 billion. Yes. So the prize money, if it is shared, you split that. Otherwise, you get it all. And then the medal. The medal is also very interesting. On the front, it's Alfred Nobel, and on the other side, it's the institution which awards the prize. So that again is very symbolic. And the diploma and the prize money. Yes.
8.9.
Yes, something like that. In terms of crores, yes. So in July, the reading of productions. In September, you have the Academy members confer, they get together, they agree on the awardee. In October, the Nobel Prize is announced. And we will be very happy to know that the 2024 prize ceremony is coming up in December, where the Nobel Prize award ceremony, wherein you give the prizes away. So this is how the process is. It's a long process. It's not an easy process of selection. There might be years wherein a Nobel Prize is not awarded, wherein it might be felt that there is no work of consideration.
No, that was for the issue of this, World War.
Otherwise also, sir. Otherwise also, if they feel that there is no outstanding work, yes, so there have been some years wherein it wasn't awarded. Wars definitely, the World War I, World War II, all those years there was no award of Nobel Peace Prize. Sorry, Nobel Prizes. It's also it is in his will.
Yeah, in the will.
In the will it is there.
There is no such. There is no such.
I think I had read wherein one particular year there wasn't. I'm not sure, but I think I read up 42, 43, and 14 to 15 World War I as well.
1941.
But apart from that, no, it says in the will, but you may be right. There may be no such a, but I remember reading in 1 year there wasn't in one category. I'll find out. So this is all about the Nobel Prize. I know I've talked a lot about the Nobel Prize because to inspire you to actually, try and attempt and, be nominated for this prize. Who knows, we might also be one, might have a Nobel laureate here. So to come to the end of my session, I think I've taken a little longer than what I should have, but I hope that you enjoyed the session. And if there are any questions, please feel free to ask any questions that you might have about anything.
Spoke about Haruki Murakami, and what I want to know is that because I haven't read her novel,
Yes,
I'm wondering
it's
that
a
Haruki Murakami is also extremely lyrical. I mean, I've read him, and it's very lyrical, and there are layers and themes, and there's a lot of intermeshing and all. Which, as you were talking about from the quotations, I get the same sort of feel, right? So I want to understand what do you think could have actually propelled the academy committee to actually for her. I mean, is that the writing style?
Yeah.
That's the tipping point.
Yes, that is because, as I said, if you remember in one of the slides, I said the Swedish Academy decided that it's only for an inventive approach to poetic prose. Right? That is the reason why they gave her the award.
But that's the same for Murakami, that's the same for
I cannot question that. I'm not in that capacity.
I
I cannot question the Swedish Academy, why they would judge, why did
No,
not
but
want
I'm
to
a
give
student
her.
of literature and I'm sure that you've read it. What do you feel? I'm just asking.
I love her work. I think whatever I've read recently, I think she's brilliant. And the way she captures, see, that is why I said that her style is very similar to Murakami. But there must be something distinct in the way that she writes, isn't it? It's lyrical, it's poetic, it's experimental.
The role of the translator also.
Translator also. His works have also been translated into English.
Right.
But the way that she has written and the way that her works have been translated, as you rightly put it, there must be something distinct about her. So with literature, I feel there is always interpretation. You might, yes? No, you might think that this author has written in a similar manner. Why has he not got the award? But as a reader, I would think that, oh, I love his work more than his work. It would appeal to me more. So it's a question of interpretation, it's a question of appeal. Literature appeals. Literature is not something which is distinct, which is a discovery. It has to have an appeal, it has to ring a bell with the reader. I might like the work of T.S. Eliot. Somebody else may say, oh no, I don't think there's anything great in his work.
See, for literature, the parameters are a bit different.
Very different.
Because they are looking for the cultural issues.
Yes.
So in Korea, on one side is North Korea, the South Korea, and culturally they are to some extent, say politically they are different.
Very different.
The
Yes.
agony and issues bothering them are different.
Yes.
And in the South Korean culture, they want to show to the world that we care for them.
Yes.
The agony,
Yes.
etc., these things.
It's there.
Same is in China also.
Yes.
Then in Afghanistan, उसको जो मिला था तो उसको मिला था किस चीज के लिए मिला था because of her interest in study.
Yes.
And they wanted to highlight.
Yes. To highlight. She's
In
also
sciences, it is different. In economics, it is again different. In economics, it is for the development of economies of the poor people.
and in fact it
So
has
whosoever working on developing the poor people, they get noticed.
yes.
In sciences, those issues which have long bearing
long bearing and impact on society. So that is why the parameters in which we interpret literature is very different. And it also is individual interpretation. Very individual. The interpretation of literature, you might read a book in a certain way, I might read it in a certain way. So it lends itself to multiple interpretations. So I'm sure when a Nobel Prize is awarded, there is a lot of, so it's not easy, it's not an easy process, there are hundreds of nominations.
There is politics.
There's politics as well. That is a section, I think everywhere is politics. See, when you writing, no. For the forthcoming novel, there are two different translators. So she has one main translator, Deborah Smith, but for the forthcoming novel, there are two different translators. So either you approach the translators or the translators think that your work is such a I would like to translate. But it has to be somebody of caliber. It has to be somebody who's going to be able to translate it so that the essence is not lost. A lot of it is said is to be lost in translation.
What it was, the hierarchy of awards and the Nobel Booker, what is the hierarchy?
Nobel Prize is obviously, but Booker is mainly for literature, for fiction. Yes, and there are so many others, but you consider the Nobel Prize. Yes, it's the same because it's at the top.
Other thing.
You have Nobel Prize, then you have Pulitzer Prize. You have the Sahitya Academy also. Our own Sahitya Academy Awards. So those are also prestigious. So that is why there are judges, and each one is prestigious. But I think this one tops it all. Yes. Sorry?
Fictional book.
It's a more fiction. It's a more fiction, The Vegetarian. And you pick up the next book, We Do Not Heart. That's the latest, which is coming in next year. So you could pick that up. I think we should. Yes, we should, because as you said, we should include the Nobel laureates. Yes. I think that's a good idea. Sir is asking us to include it in our syllabus for 2025.
Sir, ये microRNA वाला it's already part of the syllabus. जो BS.
We have the importance, sir. That importance is not realized. Know the importance. Why they were given? Because mRNA was known.
Yes.
But nobody gave it that importance which is deserved. Now they say that it has in the last slide. Because so many parameters and you can find answers, problems and find solutions. So ये चीजें जो है, these things the students they know, and teachers would know if they read it. इसीलिए हमने सोचा, I was asked कि हम स्पेशली में करवाएं, मैंने कहा नहीं, मुझे अपनी यूनिवर्सिटी में करवाएं.
Very good. Yes.
और ये हमने हर साल का करना.
We should continue this tradition. Every year we should have these sessions. It's very good idea.
Okay, so let's thank Dr. Pallavi Goswami for a wonderful talk.
Thank you so much.
I would now request Professor Kohli to kindly felicitate Dr. Pallavi with a memento.
With pleasure.
Thank you.
On behalf of all